Namaskar. The use of platelet-rich 3D fibrin scaffold as graft holding media. This scientific presentation, my research was done in 6th Latin American FE workshop at Cancun, Mexico. Last year means February 2020, just before the COVID era. I was there as invited faculty and I presented my research paper which I did. This study was done by myself and Dr. Seema at Rejuvenate Hair Transplant Center, Indore, India. So we did a split scalp study of plasma as a graft holding media. I'm going to just tell you how this study was started and how the idea of using plasma or PRP solution as graft holding means hair follicle holding media came to me. In initial days of PRP during hair transplant we used to do, and after getting the PRP solution we used to discard the remaining plasma. And one day I thought, hey, why I'm discarding such a precious solution which is biological, autologous, having the nutrients and the electrolytes which is isotonic almost. So why not to use it? And then we started putting our grafts into that plasma. And to our utter surprise, when we saw the patients on the follow-up, that grafts means the hair which usually after implantation goes in anagen effluvium, they did not go, and hair started growing. There was early growth and better quality of hair. So then we decided to do a scientific study. There are many challenges to do the scientific study. One was the global photography parameter, another was the trichoscan. We had a good trichoscan, and another important thing was I wanted to prove it histologically that the graft when we keep in plasma, and if for how many hours the matrix cells or the cells of the follicle survives. So I searched on that and I found a stain, MTT stain, which can tell me the hair follicle cells are alive or dead at that time. So that's how we started this study. So a split scalp study was done in which one side of area were implanted with the graft kept in ringer lactate. Another was the test group in which the grafts were kept in plasma and later on in the PRP solution. As I said, the parameters which I selected, one was the density which was taken at 3 months. Another parameter was the thickness of the hair at 6 months and 12 months was taken. Another thing was the global photography, and we saw the length of the hair at 6 months also, and one, as I said, was the MTT stain. This study was published in Forum International. This was also published in double-blind peer-reviewed journal of Plastic Surgeons of India, and also this study I got the research award. SAMC board Bo's most prestigious research award was given by Association of Plastic Surgeons of India, and I was given that gold medal for that. So the growth, let's discuss the parameters. We did hair density at the 3 month. It was again a surprise. The grafts which were kept in plasma on that side, the hair density was 27.5, while the grafts where the ringer was used as a graft holding solution, the density was 4.4 only. And the p-value was 0.001, which was very significant. Then the hair thickness was taken at 6 and 12 months. 6 months and 12 months, there was a significant difference, and the p-value was again significant. And histological staining for MTT stain for live cell were well stained in plasma even after 72 hours, while they were poorly stained in the ringer media. So here you can see the grafts were kept in the plasma, and here they were kept in ringer lactate, and at the 72 hours the cells were surviving. So plasma or platelet rich solution was providing the nutrients and antibodies to survive and the electrolytes for the follicle cells to survive. That is the reason they survived till 72 hours. Then anagen effluvium was noted. Up to 50 days, the anagen effluvium was not present on the site where the grafts were kept in plasma. And we noticed that length of the hair which we grew up at 6 centimeter were at 6 months were almost 6 centimeter. So these were the conclusion of the study, which proved that if we keep graft under plasma or PRP solution, the results will come early. Anagen effluvium will not be there. The hair thickness will be better. So we started continuing it. Then I searched on the literature and I realized that platelet-rich fibrin scaffold was first described by Choren et al. In France and is used for tissue engineering tool for cell culture, cell transfer and cell delivery media. The injectable fibrin scaffold improves the cell transplant survival, reduces infarct expansion and induces neovascular formation in ischemic myocardium. This study was done. Then another impressive study was the culture of the human chondrocyte to form a cartilage in vitro prefabrication of human cartilage shape using fibrin glue and the human chondrocyte. This was means entire chondral cartilage was cultured in using this 3D fibrin scaffold. Autologous culture epithelium grown in a fibrin matrix and then transferred to third degree burns. This was another impressive. These were the three studies which were very significant and proving that whatever I was doing was the worth. So the plasma used here is a platelet rich, having higher concentration of platelet derived growth factor. So in media, a fibrin scaffold with molecular signaling growth factors to stimulate the hair growth. So just understand how it works. The platelets are there. When they get activated, they have got various types of growth factors, and those growth factors stimulates the stem cells of the hair follicle, and that's how the growth we notice. And another important thing is the prothrombin, when comes in contact with tissue collagen, it gets converted to thrombin, and the thrombin converts fibrinogen into fibrin. And this fibrin is a fibrous non-globular protein. They are fibril strands which forms a network in which RBC and platelets get trapped and form a clot. Here you see in this picture how the clot formation takes place. Whenever there is an injury, the platelets, the prothrombin comes in contact with the tissue collagen and convert into thrombin, and the thrombin converts into fibrogen fibril. These are the strands, and in these strands these RBC gets trapped, and that's how the clot is formed. But when we are using PRP as a graft holding solution, means RBCs are not there, then again this fibril network fibrin forms a fibril strand network in which the grafts are kept. So each graft is surrounded by a fibrin network. So it's a scaffold. So it's a 3D scaffold around a graft, protecting the graft from dehydration, desiccation and microtrauma, and providing the nutrition. Another important thing is platelets were also trapped in that 3D fibrin scaffold, and these platelets are constantly releasing the growth factor, and this growth factor works on the stem cells of the hair follicles. So this you can see, this is the scaffold. Some finds it's a side effect or the drawback of using the PRP solution as a uh, platelet as a graft holding media, but I tell you that's what we want, because we want that the graft should be surrounded by this fibrin mesh. So this is a basically this engineering module in which the hair follicles are there, cytokinins and growth factors from the platelets are there, and a 3D fibrin scaffold which is surrounding the hair follicle. It protects and stimulates neogenesis, angiogenesis, and epithelialization, and that's what we want. This is another innovative things done by me. This is a very specific platelet rich solution forming kit which I made myself. I went to the glass factory and made almost 10 models of it, and finally I finalized this, and this is an autoclavable and gives me the good concentration of platelet. This is the scheme how we do it, how we get the PRP solution, how we get everything. This is the solution we get. Fibrinogen gets into fibrin mesh and platelets gets activated and they release the growth factors. Now you can see this video. We take out the blood, we transfer this blood into these two vessels, put them in a specialized centrifuge machine having the temperature control. First we do the separation of RBC from the plasma, then plasma is taken out and put in another glass tube, and second spin is done for the concentration. So platelets sits at the bottom. This you can see beautifully. Platelets are sitting at the bottom. Then we make a PRP solution out of this, and this PRP solution is used as a graft holding media. This is the PRP solution we are going to use, and we have kept the grafts into it. This forms becomes like a gel. See how beautifully these grafts are swimming in the PRP solution. Then these grafts get stick up. This is a problem, but by learning we can separate these grafts very easily without creating the trauma to the delicate graft. So now let's see the steady results. At 35 days we saw anagen effluvium was not there and the hairs were growing. That was the 35 days. We did both sides steady, means both sides the grafts are put. This is another at the 50 days. You can see here. This is at the 60 days. Still the hairs are growing. So there was an early growth. See, at 90 days, almost 2 centimeter, more than 2 centimeter hair growth we could see just after the hair transplant. This is 116 days growth, which looks quite good. You can notice the quality of the hair. This is 4.5 month growth. Good length has been achieved of the transplant. This is another at the 5 month, almost full grown growth with good quality of hair. Then another point was the quality of hair, means thickness and the shining and the glow. You can see how thick they looks and healthy and having the good glow. So that's the quality also improved. You can notice here the density, quality and the thickness of hair. This is another case. So the conclusion is 3D fibrin scaffold with platelet growth factor is a good graft holding media to prevent or control post-transplant anagen effluvium, early hair growth and a better quality of hair with more density of transplanted hair. Future is preparing to use the platelet lysate to deliver more growth factors and follicle-derived stem cells to boost up the growth. So this was an incidental finding, and then we made a scientific study and we proved that platelet rich plasma or only plasma, platelet poor plasma can be used as a graft holding media, which will give you better result in terms of early growth, in terms of reduction of the anagen effluvium, in terms of better quality of hair, and it is not difficult. No extra cost is we have to pay for that, and it's very easy and anyone can do it. Thank you very much. Thanks. So you put extra fertilizer to get a good growth of plant, and that's what we did here. Thank you again.